Hey friends, so there have been some questions about the homework, and I thought I would address it in a little video discussion. We kind of went through this a little bit on Thursday's lab, but just for everybody. So, the goal is to hand translate all 6 frames of a given DNA sequence. First, let's just go through how we achieve the frames. Here is a given DNA sequence with a crappy marker. All right, let's see. Where's a better marker? All right, so there's a sequence. So when you're given the sequence, the forward reading frames are simply taking that sequence and using the first 3 nucleotides as the codon, second 3, third 3, and you find the 3 different reading frames. So you have GAA, TTC, AAG, TCAG. The second reading frame, you start one in, and it's going to be AAT, TCA, AGT, CAG. And this is why you write it on the notebook paper the way we showed it, so you can actually arrange things. And it's a good idea to sort of group them, in groups of 3. And the third reading frame is ATT, CAA, GTC, AG. Okay, so once you have the 3 reading frames, you then go through and decode it. You identify the individual amino acids. To get the reverse reading frames, you do the following 2 steps. First, you complement the sequence. So that would result in the following: CTT, AAG, TTC, AGTC. Okay, so you complement it, and then second, you write the sequence backwards, i.e., you reverse the sequence. But you got to do these 2 steps. So the reverse would be starting here and writing it backwards. CTGACTTGAATTC. That is now the reverse reading frame. And you simply go through and identify the 3 different reading frames. Okay. So now once you have the 3 reading frames, the next step is to blast these reading frames. And that takes you to NCBI, the National Center for Biotechnology Information. Now, let's see. All right, we're getting our video up. The room is kind of bright. Hey, Igor, can you close that big curtain? Just you have to reach back there between the 2 specs and roll the plastic rod. It's a little bit of a lean. All right, we're going to darken the room a little bit more. That's perfect. All right. So, sure if you want to. Very good. Now, this is NCBI. Let's see. So if you go to that link, I think it will bring you to this spot. If you go to ncbi.gov, go into resources and go to DNA and RNA and hit blast. All right. So this is, I think, where the URL I gave you will send you to, and you go to protein blast. Bip. Now, a question was, it is set for looking at small sequences, so you don't need to do any clicking. All you got to do is enter the single letter abbreviation code. Let's see, I may have one. I didn't want to tell you about my visit. Thank you. Yeah, there's an amino acid sequence that I actually pulled up for one of our vitamin C projects. And all you got to do is put your sequence in and hit blast. It will optimize for the size of the sequence that you got. You run through, let it go, and depending on how busy blast is, it takes a while, and we're waiting, and we're waiting. So if there's a stop codon in the middle of the sequence that you're doing, then you don't have to blast it because there ain't going to be one. When you're looking at the different frames, if you've got a only two codon or two nucleotides at the end, that's not full codon, and you just don't go there. So only blast those sequences that have a sweet open reading frame, and you come up with this. Over the years, they've changed the blast page. This is the only page you need to print out, the first page. You can also do a graphic summary where if you put your mouse on it, it'll tell you what the individual gene is. In this case, we're looking at a portion of the enzyme that's involved in vitamin C biosynthesis, which is called galonolactone oxidase, and we've got a lot of different sequences. You can also hit alignments, and it shows you how well they align and the individual species. And so you'll be able to identify, hopefully, the gene that you pulled up and go down that list at the bottom of the page, and you should find a matching gene. So that's how you do this. Blasting is simply a matter of putting the sequence in and hit and go. It'll work, okay? Just for your own purposes, there are databases you can choose to search. We're using, and the set standard is what's known as the non-redundant protein sequences, which is just all the different protein sequences they have. If you hit this arrow, you can see that there's what's known as the RefSeq. This is the reference set of proteins. There are model organisms, that things like flies and mice. There is patented protein sequences, which I've actually not explored before. A variety of different things. Just stick with what is set up, and you're ready to go. It's a way of narrowing the search. And you can exclude organisms or include organisms, and there you go. But the algorithm program selection is set. BLAST-P, protein, blast is exactly what you want to do. Hit it and go. All right. That's how you do this. As I was mentioning to one of the labs, there is a program called XPASI that will actually translate everything for you. And let's see. Yeah, there's XPASI, and it's xpasi.org. And or if you just Google up XPASI, EXPASY. Here you can put the DNA sequence in and say, translate it. And bing, it translates all of the 6 reading frames. So you got the 3 forwards and the 3 reverse. And then what you can do is pick one of the open reading frames that you like, or pick a sequence and go. And there you have it. All right, that's cheating though, but if you want to check your sequence, you can do that. See how that goes. All right, that's all I have to say for that. And if you have any questions, hit me up. I'll try and help. I'll be talking with you later. Bye.